So I do want you guys to understand and be able to compare and contrast. This is brighter in here than it only does. Compare and contrast the way that bacteria and plants make it versus mammals. Because anytime, and this is why I tried to scare into the nurses, with medicinal chemistry or doctors or anything like that. That my other classes, anytime that you can exploit the differences, you have a potential drug, right? Because it's something that's essential. We could use this as a possible drug if we could have a way to knock out bacteria versus leaving humans alone, so on and so forth. So it's always nice to have an idea of the differences between the different classes, not biology, the different organisms. Okay? And so here I didn't even hide I don't think I even hid the boxes here just to know the basic steps, and there are certain questions to really understand. For example, the big difference is where does the sulfur come from with respect to the cysteine? Bacteria and plants get the sulfur from one thing, and we get the sulfur from something else. Okay, and so because that's what makes a cysteine, like the reactive thiol. So just know the basics on this one. Ultimately, if you go back far enough, they're going to get their sulfur from sulfate. So there's lots of sulfates out there. Now notice one thing that's similar to us, that serine is used. So that's why this one looks like a two-pronged attack. So we have serine, and the name of this should make sense, called serine acetyltransferase, because you're adding an acetyl group to the serine. That's no biggie in the enzyme. Okay, but here it's a sulfur. The sulfur actually a sulfurylase, which that's the first time, to my knowledge, that may be the only time this semester that we see this, but that is transferring a sulfate. Okay, and so that's why we now have this APS instead of ATP there, and then that sulfate gets, it gets phosphorylated once again, and I would never ask you to be able to draw these or anything. The important thing is, first of all, it uses serine, but it's that the sulfur comes from sulfate. I can't stress that enough. There's a reductase that ultimately takes the sulfate down to a sulfite. Then the reductase takes it from sulfite to sulfide, which I would expect you to be able to look at the differences and understand that it's oxidation going this way. Anytime you're increasing the sulfur to oxygen bonds, you're oxidizing it. Anytime you're decreasing those, you're reducing it. Okay, so then we have a sulfide, okay, which is an anion. And that sulfide is actually through a O-acetylserine thiolase. It's not thiolase. Okay, it's a thiolase. Okay, and that's how it makes cysteine. So once again, to reiterate, the amino acid body portion of cysteine comes from serine, but the sulfur comes from sulfate. Okay, to make sulfide that gets incorporated into the cysteine. Now, the way humans do it is different. Now, I want you to know the details of that. Most of you are going to be interested in human medicine. Once again, serine is used, but serine is not the only amino acid that's used. Did I tell you where that come from? Oh, I will. Okay. There's another amino acid. It's called homocysteine. You guys remember, we've talked a little bit about homocysteine. I think I mentioned it briefly. I usually use it as an example of an amino acid that's not used to make proteins in the first semester of biochemistry. But you guys know, like homocysteine? It looks a lot like cysteine, but it has an extra carbon. See, it's got an extra linking carbon. So it's called homocysteine. It even has its own three-letter code, which is HCY. I don't know if it has its own one-letter code. I don't know it. So I want to ask you guys to know that. But sometimes you will see it abbreviated as HCY. It's important. I mean, obviously it's important in this metabolic pathway, but it's also important in the upkeep of your heart. And so I usually also use that as an example for metabolism for cardiac. The woman who discovered some of the uses, the biomedical uses for homocysteine, her lab, first of all, she was on my board for my PhD, and her name is Rowena Matthews. I don't know if she's still practicing or not because, I mean, she would be a very mature lady now, be getting up in years at that point. But she actually was named to the Institute of Medicine for this, which is, in science, the highest honor you can get in the United States. There are two, and they're of equal footing. One's called the National Academy of Sciences, which is over all sciences, and then there's a portion that's called the Institute of Medicine, the IOM, and so she's named to that. And so it's almost like being knighted, if you want to think of it that way, in science. And so it's a high, high honor. And in theory at least, the National Academy and Institute of Medicine are supposed to be the body of scientists that help advise Congress and the president and anybody that asks whenever they talk about bills and things like that. And so Marina Matthews actually was honored to be put onto the IOM because she discovered that homocysteine with homocysteinemia and homocysteinuria. Homocysteinemia, of course, means it's elevated in what part of your body. Levels are elevated. With homocysteinemia, it's in your blood, and homocysteinuria would be urine. And what happens is it's an indicator of heart disease, okay? Cardiac issues when you have, especially in your blood, if homocysteinemia starts occurring. And later, let me see, it may be sooner rather than later. Yep. Sooner rather than later, we're going to discover the reason why. Because homocysteine is actually metabolic intermediate for a number of pathways as well. But again, for us to make cysteine, we can make serine, like we've just been discussing on Friday. And then we use PLP, which is pyridoxal phosphate, which we've seen that before. And PLP is a cofactor, and it's going to take the homocysteine and the serine and combine them to make what's called cystathionine. So what class or classes of enzymes do you suppose this can be? It's a synthase. How do you know it's a synthase and not synthetase? No ATP. So if you were going to name this one, what would you name it? Say it with some conviction. Yes, this is cystathionine. They could say beta, synthase. And the only reason why it's beta is because it's telling you what carbon it's linking to. It's not linking it on the alpha carbon, beta carbon. But if you tell me it's cystathionine, synthase, I'll give you the points for it. See, once again, not too difficult. Then, essentially, all we're doing here is we have this enzyme that's also using PLP as its cofactor, and it's chopping or breaking that bond between the sulfur and that carbon. So you're going to get alpha ketobutyrate, which this in of itself is an alpha keto acid. Okay, because see, there's the carboxylic acid, alpha keto acid. It's just one that's a different one than what we've seen before. But we can use alpha keto acids for various reasons. If they get out of whack, then that's bad. But okay, and we get cysteine. Does anyone remember what class of enzymes are used to make or break like carbon-carbon bonds, carbon-sulfur bonds, and carbon-nitrogen? What? It's a lyase. And so if we were going to name this one, what could we name it? This is cystathionine, and it tells you, again, it's technically a cystathionine gamma lyase, because it let you know which bond was broken. But if you had just told me cystathionine lyase. Lyase, for me, was always one of the classes that was hardest. I'd always forget about, because we didn't see it very often. But it is a lyase. And so we make cysteine. So one of the big difference, first of all, if we're comparing and contrasting bacteria and humans, what's one thing that they both have in common? What's the amino acid that it both originally comes from? Serine. Now the big difference, once again, is bacteria and plants get their sulfur from sulfate. We get ours from homocysteine. Now homocysteine comes from something else, somewhere else. Does anyone know what amino acid do we get homocysteine from? It's not from cysteine. Because you wouldn't want to make make a cysteine, make a homocysteine, make a cysteine back.
Would it be methionine?
It's methionine. Because what's the only difference, if you look at homocysteine, what's the only difference between homocysteine and methionine? The extra methyl group. It has that S methyl group, okay? And so homocysteine actually comes from methionine. So we get our sulfur from methionine. Bacteria and plants get their sulfur from sulfate, which is used to make sulfide. Okay. And this is where our good friend S-adenosylmethionine, SAM, it's also called Adomet, comes in. It's an important cofactor. And homocyst is homocysteine. I got sloppy. I was typing it all up. And as I mentioned just briefly before, homocysteine is linked to a whole host of medical varieties. Many of them, it's a major indicator. I think it's pretty common now whenever they do cardiac panels and things, often if you have cardiac issues that they will now check. And that's actually more recent years that they'll check for homocysteine, because it's an indicator that you have something messed up with metabolically. So now we're going to look at this. I haven't covered these names because I really want you to know it's technically from chapter 18, so it would be a review, but I'm sure that you don't remember it. Maybe you do, or maybe it looks familiar now. But this is how we get it, this sulfur to make the cysteine here. As we start with methionine, and sulfur is a nucleophile, as we can see here, because it's got the lone pair of electrons, and that's the methyl group we've got to get rid of. Usually when we're talking about Adomet and everything, the important part here, of course, we have the methionine adenosyl transferase to make SAM or Adomet, whichever we want to call it. This is used many times for methyltransferases, and that's what we talked about it back in chapter 18. We used it for methyltransferase, which is where they were transferring that methyl group. But we're also using it in order to get the homocysteine. That's another possibility. So once you have SAM, all these, there's lots and lots, and for those who've taken chemistry of cancer, we've talked a lot about methyltransferases. This is a class of enzymes that will use SAM, and they will actually literally use up that methyl group to add it on to whatever needs to be added on to. Then we have, instead of Adomet, we have S-adenosyl homocysteine, which I think they may abbreviate as SAH. Okay, but then there's a hydrolase that comes in, and water comes and smacks off the homocysteine. Now, if we're going to repeat this cycle for methyltransferases, then what you're going to do is use your tetrahydrofolate in a synthase in order to make methionine back. But if we need to make cysteine, we can utilize that homocysteine now with the method that we just talked about, okay, in order to make cysteine. Sorry. All right. So now we're going to change here. So remember, we're going through all the amino acids. We're going to be talking about alanine, aspartate. Just briefly, which should be review. Where does alanine come from? Pyruvate. And what's the enzyme that's used? The aminotransferase. What one in particular? Not pyruvate aminotransferase, but alanine aminotransferase, because it's usually named after the amino acid. And aspartate comes from oxaloacetate, and the enzyme aspartate aminotransferase. And so there, we talked about those. So now we're going to move on. So now we have asparagine. This one, you probably can figure it out, or figure out the basis of it based off of what else we talked about so far. So asparagine is a lot like what other amino acid? There's two possible answers. Asparagine is a lot like aspartate, and it's a lot like glutamine, right? Where did glutamine, do you remember? Because we've already talked about glutamine. Where did glutamine come from? What amino acid made glutamine? Glutamine. So, through your hypothesis, based off of our two things, where do you suppose that asparagine is going to come from? Aspartate. Sorry, this was the only picture that I blatantly plagiarized off of Google. But I forgot to cite it. So this actually takes you a step back. It's always a good review. So we're going to take a big step back and just review this. When we have oxaloacetate, what is this right here? What is that player? That's glutamate. Okay. And so glutamate becomes, what does it become right here? That's alpha ketoglutarate, which I'm just going to abbreviate as alpha Kg for space. So what enzyme was this that just did all that? Pardon? It's an aminotransferase, and what one in particular? It's aspartate. Because we just made, I'm just going to call it asp. Okay, because we made aspartate. So what is this right here? It may look a little odd, but we've seen it before. Gluta what?
Glutamine.
That's glutamine. Because it's got that amide, that terminal amide. And glutamine is being turned into, what's that product? Number 5. Glutamate. Okay. Now this one, if you notice, we have ATP. This is, it's actually more than one step, but they're showing it as just one step because it's one enzyme that does all of it. We have an ATP that's being turned into AMP, an inorganic pyrophosphate. So what class or classes of enzymes could this one be? Is it synthase? Synthetase. Because it required ATP. And this one is asparagine, which I'm just going to use its 3-letter code ASN, but it's asparagine synthetase. So once again, if you just take some time, which I realize sometimes on exam days, so much. Don't overthink it. Okay.
So you said that was more than one step?
Yes, anytime, almost always when you see the ones where it says like ATP going to make, even if it said ADP and phosphate, you can, the whole purpose of the ATP there is that it activated some intermediate. Mm-hmm. So if I had to hypothesize on the mechanism for this one, I would say it was probably first amylated, and of course AMP is just a great leaving group, and so then the nucleophile, which would be the nitrogen in this instance, would be able to come in and take it off.
You wouldn't have to know the different steps, just know.
Nope. Mm-hmm. Nope, not unless I specifically asked you to know that mechanism, which we do have a mechanism coming up soon that I just wanted to show you because it uses a different type of catalytic triad. Okay. This is just changed. Okay. So asparagine can be degraded to form aspartate. Just like we saw glutamine that can be used to make glutamate. This is just another way to make aspartate. Does anyone know what? Whenever we did that, does anyone remember what was the name of the enzyme that transformed glutamine into glutamate? It had a common name. Glutaminase. So, knowing what you know, what do you suppose the name of the enzyme is that will take asparagine and make it to aspartate by deaminating it? Not aspartase, but what was the original one? What? Glutaminase. Asparaginase. Right? Asparaginase. Right? Yes, asparaginase. But this one actually has a double catalytic triad. If you think, and I know we covered this part because in when we did enzymes, which has been a long time ago. And triads are very frequent, or frequent players you see over and over again. But remember when we did the proteases, we had that catalytic triad, which was an aspartate, which would take the hydrogen from a histidine. So then the histidine would take the hydrogen from usually a serine or a cysteine. So that way it just makes it a better nucleophile. This one actually has 2 sets. And so I was going to just point it out. And I would never ask you to draw out the entire mechanism because of the fact that it's in depth. But I would expect you to be able to recognize, remember what a general acid versus general base could do, and make hypotheses based off of, if I gave you the mechanism, make hypotheses based off of, if there's mutations here, what would it have, which one would be better or worse, or that kind of thing. All right, let me make this smaller. This one's very different. So this one I got from a paper, and I really wish I would have put that on there. This is asparagine. Remember, the whole point is we're trying to get rid of the nitrogen. And this threonine here is just holding it in place. It's not part of the triad. The triad here is a glutamate, a tyrosine, and a threonine, which we haven't seen threonine really do anything like this before. We've seen tyrosine with tyrosine kinases go out and be nucleophiles, but threonine, it's an alcohol. It's just kind of in odd shapes. Serine is the one that we've seen the most. But is the glutamate right here, is it acting as a general acid or a general base, if we think back to last semester? If it's taking the hydrogen from the tyrosine, is it an acid or a base? It's a base. It's acting as a general base. Okay, so therefore the tyrosine would be general acid right there. And by the way, the tyrosine is going to be taking the hydrogen from here, which makes it a general, it does both. The triad that we looked at last semester, where it was like histidine. Remember, one nitrogen, the histidine was giving up a hydrogen, while the other nitrogen, the histidine was taking it? Okay, and the only reason why you do this is because now it makes that threonine act more like if you remember, it's called I'm thinking of the word, a better nucleophile, because it's acting like a alkoxide. That's what I was looking for. It's not completely an alkoxide, but we go and we smack that, boom. Of course, that's going to pop up to make the tetrahedral intermediate, which is what we've seen here. And just like what we've seen time and time again from organic chemistry, when you make that tetrahedral intermediate, you're going to collapse it or break it. And so, boom, something has to break or leave. And the great leaving group here, or not great, what's become a better leaving group here is that it's going to leave. There's the nitrogen, it's going to leave. Okay.
Do you want us to know, which one's active acids and bases?
You could. And just remember, an acid is going to be donating the hydrogen, and the base is going to be accepting the hydrogen. Or I could ask what's being a nucleophile. Well, here, in this step right here, it's going to be this oxygen that's the nucleophile. So one question, for example, is if I gave you a choice of questions where this threonine is being mutated to various amino acids, what's one amino acid here that if this threonine was mutated to it, that would probably have little effect? It really wouldn't matter as much. Serine. Serine would be relatively safe, Whereas if I had given you aspartate, that probably wouldn't be as good, okay, because the carboxylate's not nearly as good of nucleophile. If I had given you phenylalanine, that would be really bad, right? Or arginine, or something like that, which is something completely way off left field. All right, so then we just get collapse, and then you release that ammonia. Oops, now let me go back down because it made it really big. Then step 3 is when the second catalytic triad comes in, and it's going to activate a water. Okay, so this is where this paper, one thing I don't like about it is it kind of shifted stuff around. But here's what had been the asparagine that's going to become the aspartate. But now it's a different catalytic triad. And see, right now it's the water that's the nucleophile. But water by itself, it's a decent nucleophile, but it's much better if it's been activated where it behaves more like a hydroxide, right? And so the way that it does that is we have a lysine, an aspartate, and a threonine. Okay, the lysine, or actually, I'm sorry, let me start here with the aspartate. The aspartate is actually taking one of the hydrogens, or can take one of the hydrogens away from the lysine, because then the lysine can then coordinate to the hydrogen for the threonine. The threonine is going to take the hydrogen from the water, and that's just going to make that water much more easy to attack. You're going to form the tetrahedral intermediate, just like you did before. You're going to collapse that tetrahedral intermediate, and whenever it collapses, the leaving group now isn't the OH, it's the original threonine. That's the leaving group, so that's how you get your enzyme back. So once again, that's why I said I wouldn't ask you to draw this all out, because it would take a while to draw out 6 amino acids in water and an asparagine.
So I'm guessing this is reversible, right? The OH could just leave and go backwards, or is it not reversible?
At this point in time, it would be reversible. I don't know in vivo if the entire thing is reversible, because one thing that happens, which we didn't go into in enzymology, we would in enzymology, but we didn't last semester, is sometimes one of the products, which would have been that nitrogen that first time, or possibly even, probably the nitrogen this first time, when it leaves, it's almost impossible for it to come back, because the odds or the likelihood of it coming back are so small that it makes the entire process essentially irreversible. Okay, but an individual step can't. At this point in time, it could. That water could leave, most likely, if I had to hypothesize. But it's probably the driving factor to go it forward overall will probably be the nitrogen leaving. Especially in vivo. We're not going to have free nitrogen floating around, because we already know we don't want nitrogen to stick around for long term. Okay, but then whenever this leaves, we get our aspartate. See, now they're showing it being hydrogen bond still in place in the enzyme, but we have our threonine back. We've got that original threonine. And so all is good, and the aspartate would leave. But remember, that enzyme is called Asparaginase. What happened? All right. Of course, I always like to kind of tie this all in together to some medicine at some point in time. So asparagine actually has metabolic implications. I already kind of mentioned homocysteine anemia and homocystinuria, but now we're going to talk about asparagine. I try to say asparagine and when asparagine goes out of control. Well, okay. Sometimes you just point. Okay. All right, so for whatever reason, and they don't know a lot about this, but there are types of acute lymphoblastic leukemia that requires asparagine for growth, and so you see an uptick in asparagine. So if, knowing this, what's one possible treatment? If, in this, specific for this kind of juvenile cancer, if you know that it's going to require asparagine in order for that tumor to grow, what is a good treatment, do you suppose, for ALL? What's one way? Okay, one is to limit asparagine from the diet. Of course, one of the problems with that is the fact that we can get, asparagine is not an essential one, because we've seen we can get it from aspartic acid. What's another way that we can think about here with what we just talked about? Instead of limit the amount that they take in or make, what's another way that we can do it? Break it down. If there are ways to break it down, if we can upregulate breaking it down to where it doesn't stick around long enough for it to feed. At least that could be a nice, what's the word I'm looking for? Not cofactor, but co-treatment. I know there's a word that I'm looking for and I can't think of it. Adjuvant, isn't that the word? Adjuvant? For therapy, to where you could have it side by side with something. And lo and behold, they've done this. They actually just administered, along with the chemotherapy, they say, okay, what if we just give asparaginase? We just give the child here extra, something that's natural. Okay? And what happens whenever they do this is more than 95% remission rate. Now, unfortunately, 10% does suffer from relapse, but it's still pretty good. So this is just one way to show that you can manipulate things that occur naturally to help increase the likelihood of success.
What are the implications of decreased asparagine, like for the rest of the body?
So one thing is, would you want to look at, how would you deliver this? Are we delivering it orally? PO. Because then it could be systemic, or is there a way? I mean, I don't know. I don't know. My guess is they probably do deliver it orally, or maybe through IV or something like that, in order just to where they're more worried about getting rid of the tumor, having a better success rate for the chemotherapy or the other phototherapy, whatever it is that they're using for that tumor. And then long term, saying, okay, we can have them decrease asparagine elsewhere. But you could try to look into ways to where you deliver the two together, localized. So now there are lots of different methods that they're looking at, ways to covalently attach, enzymes and antibodies and things like that, to where it gets delivered specifically to the site, or maybe just if the tumor's big enough, injecting it in situ. And then current studies are looking at asparagine synthetase inhibitors, right? So that's another way. Just if we limit the amount that your body naturally makes, can possibly help increase the likelihood of the other types of chemotherapy being successful. Are there any questions? That's